Hi everybody. For today's lessons we're going to be learning about karyotypes. Remember that different organisms have different number of chromosomes. For example, in a typical diploid human cell, like a skin cell, a bone cell, a blood cell, you're going to find 46 chromosomes. Alright? Now, that varies depending on what species you're looking at. So 46 is the diploid number for humans. In a dog, you're going to find 78 chromosomes in one of their diploid cells. In a cat, you're going to find 38 chromosomes. Right? So it varies from species to species. Right? But humans, we have 46 chromosomes, right? Which are arranged in 23 pairs. Right? And we see the 23 pairs of chromosomes arranged right here in this picture that's called a karyotype. Now, the first 22 pairs of chromosomes are called autosomes, right? They're the same for both genders, males and females, right? It's the last pair of chromosomes that are sex chromosomes, and we've already learned about them. This particular karyotype, we know, is from a male because they have one X chromosome and one Y chromosome. If it was a female, they would have two X chromosomes and 0 Y chromosomes. Okay. So, a karyotype is a photograph of the complete diploid set of chromosomes grouped in homologous pairs and arranged in order of decreasing size, right? So, chromosome pair number 1 is going to be the pair of largest chromosomes, and then chromosome pair number 2 is going to be slightly smaller, and then 3 is going to be slightly smaller than that. Right? Our 22nd pair of chromosomes are going to be our smallest pair of autosomes, right? Now, it gets decreasing order until we get to the X and Y chromosome, right? That's the one exception. Your X and Y chromosomes are always your last pair, even though your X chromosome is a relatively large chromosome. So, how to make a karyotype? Step 1 is to photograph a cell that's going through mitosis. Again, we want to analyze the chromosomes. So why couldn't you photograph a cell that's in interphase to make a karyotype? And the problem with interphase is that chromosomes aren't formed during interphase. Right? The chromosomes are all uncoiled and they're kind of like loosely packed mixtures of DNA protein in the nucleus in the form called chromatin. Right? So after you photograph the chromosomes, you then have to match them up, and this can be quite tedious. We know that homologous chromosomes have a few things in common. Right? Number 1 is that they're always going to be the exact same length. Right? So this homologous chromosome and this homologous chromosome are going to be the exact same length. They have the centromere, this connecting point, at the exact same location. So for this particular chromosome in the center, it doesn't have to be in the center though. It could be closer to the top. Right? We're all the way at the very, very top. Right? The last thing, and probably the most important, is that homologous chromosomes will always have the same banding pattern. Alright? So, these areas of light and dark, it's just parts of the chromosome that pick up stains in different ways. Right? But homologous chromosomes are different in the alleles. Remember that you inherited one of these chromosomes from mom and one of these chromosomes from dad. So even though they contain the exact same genes, they may have different alleles. So at the top here might be the gene for like hair color, but this is the allele for blonde hair and this is the allele for brown hair. Alright, so the genes are the same, but the specific alleles might be different. Alright, the last step of making a karyotype, so let me go back a slide, is just to arrange the chromosomes in order from largest to smallest. Again, X and Y chromosome is your one exception. Right? So here we have an example of a karyotype. Right? And there's a couple things that we can learn by making a karyotype. It is a tedious process, but a useful process, right? By looking at this karyotype right here, we know that we're not looking at a human being, right? Because they only have 40 total chromosomes, not 46 chromosomes, right? So, whatever this organism is, it's not a human, right? We do know its gender. You see, it has one large X chromosome and one little Y chromosome, making this thing a male. Alright, and then we can see if it has any chromosomal disorders, alright? You're looking for any chromosomes or parts of the chromosome that are missing, or anything that's extra when you're looking for a chromosomal disorder. And if we scan, everything looks like it's matched up correctly. We don't see anything missing. We don't see anything duplicated. There's probably no chromosomal disorders in this organism. Alright, so that's why you would want to make a karyotype. We'll actually continue talking about karyotypes and chromosomal disorders a little bit on in our later lessons this chapter as well. But that's it for today. Thanks for listening, guys. I'll see you later.